All right, good morning, ladies and gentlemen. Let's get started. So we started a discussion, today's Thursday, we started a discussion on Tuesday about the kidney and how we're going to look at that really as a volume regulator. So we've got two sets of tubes that we need to think about. We need to think about that vasculature that is hanging out around the nephron, and then we need to think about the nephron itself. Okay, so there's two sets of tubular systems down here that we're going to be exchanging volumes in between. Okay, so we started thinking about the control of exchange between the capillary bed, one of the capillary beds of the kidney, the glomerulus, and we'll see in a minute where that fluid flux will be going into the beginning of the nephron, the beginning of that tube, the Bowman's capsule. Okay, and we know from understanding fluid flux within any other tissue that pressure in the capillary, hydrostatic pressure in the capillary, or in the case of the kidney, hydrostatic pressure in the glomerulus is going to be a big driver of the way fluid moves. Okay, so we looked at the arrangement, the special arrangement that the glomerulus has, the capillary bed, the first capillary bed in the kidney, has with arterioles, right? Special relationship with two sets of arterioles, so we saw some pretty slick control of capillary pressure here, or pressure in the glomerulus, that we hadn't seen in other systems, okay, because we put a capillary bed between two arterioles. So now we are in control of volume into, but we are also in control of volume out of this compartment because of those resistance arterioles, okay? So the body is going to utilize constricting these in different ways depending on what needs to have happen at the level of the kidney with fluid movement. Okay, so we will see as we move forward that both afferent and efferent arterioles are differentially controlled. Sometimes we control them together, but sometimes we control them separately in order to be able to change pressure in the glomerulus, in order to be able to change fluid flux at the level of the kidney. Okay? So that's really where this piece fits in terms of our big volume regulation control, especially when it comes to then thinking about mean arterial pressure. Okay, so that's where we were at the end of lecture on Tuesday. So now I'm going to think about control of that vasculature before we leave here. So thinking about how do you control arterioles. Okay, so we're going to see some similar mechanisms that we've seen before. So the control of the vasculature, and what we're talking about here is that resistance vasculature. So we are going to talk about afferents and efferent arterioles here. So these are classic arterioles. They have smooth muscle on them so that when you stretch them, they will constrict. Okay, so they have that classic myogenic response. So this is a vascular bed indeed that autoregulates just like every other vascular bed that we've seen. And that autoregulation and that myogenic response, we've talked about it now. We talked about it when we looked at the gut. We talked about it when we looked at arterioles in the rest of the vasculature. Talking about it here again. Autoregulation is going to keep flow constant over a range of pressures, right? And how is it doing that? By changing resistance. Okay, so I'm gonna write it down again, but just note that we've been here a couple of times. So these aren't special in terms of the myogenic response. So they do autoregulate, keep flow constant. I don't know what that word is. Going to keep flow constant over a range of pressures. So we've seen this graph before, over a range of pressures or in a normal physiological range between 50 millimeters of mercury and 200 millimeters of mercury. When pressure increases, the blood vessels will respond by constricting in order to keep blood flow constant. And in this case, we are thinking now specifically about renal blood flow. So this really, if we say the graph, or if we think through the graph in words, this really means that, for instance, an increase in mean arterial pressure results in an increase in resistance, a decrease in radius, therefore flow remains constant. Okay, so if we think about flow equaling P1 minus P2 over R, we increase pressure, we increase P1, we should expect flow to go up, but it doesn't because something happened to resistance. Okay, resistance went up as well, so if the numerator goes up and the denominator goes up, flow stays constant. Okay, so that's essentially what happened with why flow remained constant. So the question then becomes why, when pressure went up, did resistance go up? And that's that myogenic response, right? Smooth muscle, when you stretch it, it constricts. We have no idea what that mechanism is, but it's pretty consistent over smooth muscle, overall smooth muscles in the body. So just as a reminder, it's the myogenic response that's changing resistance. Changes resistance in response to a change in pressure. So stretch causes contraction. That's really what, physiologically, that's what we're talking about. It's really important at the level of the kidney for us to be doing this. If we change pressure, obviously based on this relationship, if we change pressure, right, that mean arterial pressure, we change volume into, we have an ability to change pressure in the glomerulus, right, just by changing that, just by changing mean arterial pressure. So you can change mean arterial pressure at the level of the kidney every time you stand up. So when you actually leave from class today, when you stand up, your blood volume will be drawn into your legs based on gravity. That's going to be an increase in pressure at anything below here. Okay, so that you would have just experienced an increase in pressure to the kidney. That would, if you didn't go through autoregulation, you would have an increase in flow to the kidney and an increase in glomerular pressure. And we'll see in a minute that means you're going to filter more. Okay? So depending on then how many times you sit down and stand up in a day, you could be filtering like crazy. And remember, we're sending 1.2 liters of blood to this kidney per minute. So the ability to lose blood volume by sitting and standing in a day is really high, right? But the good thing is that when you stand up and you increase that pressure to the level of the kidney, stretch causes contraction. So flow stays constant. So now, no matter how many times you sit and stand in a day, you're not going to change pressure in the glomerulus based on that maneuver, and you're not going to lose excess amounts of fluid. Okay, so that's why we have the myogenic response. Try to keep flow constant because we change our body position all the time. Okay, we don't want to be fluxing fluid based on body position. Okay, so the myogenic response lives at the level of the afferent and efferent arterioles. So on top of that, there is a control of radius of these resistance vessels, okay? We do have direct SNS innervation into afferents greater than efferents, okay? So SNS onto a vascular smooth muscle will cause constriction, so that's the normal, right? What's a little bit different here is that it's going to cause constriction of afferents greater than efferents. Okay, and that gets really critical now. We're starting to play a game, right? So the sympathetic nervous system is going to constrict upstream of this glomerular capillary bed greater than downstream of this glomerular capillary bed, right? So this maneuver is going to change pressure in that capillary, and therefore we are going to change filtration. Okay, so anytime that we're going to change them separately or differently, we are going to get a change in pressure in the glomerulus. Vasoconstriction of afferent over efferents, right? We're going to have less volume into, more volume, as opposed to the volume out of. If I have less volume into, I have less volume in, less pressure in. Okay, so that's kind of a common argument that we've been playing with a lot. So that maneuver will result in a drop in pressure in the glomerulus. So we've got nervous control, direct SNS innervation. So not only that, whenever we see SNS control, we should also be registering now that we can do this very quickly. This is a milliseconds to seconds maneuver that the body can use in order to control pressure in the glomerulus. Okay, so we can do it quickly. So we also have local autocrine and paracrine control right at the level of the kidney. So nitric oxide is a vasodilator, but it vasodilates both afferent and efferent equally. So if locally we were to expose afferent and efferent arterioles to nitric oxide, we would have no change in pressure in the glomerulus, okay, because we change both ends. We'll change renal blood flow because we changed both ends, but we're not going to change pressure in the glomerulus. Endothelin 1, vasoconstrictor of afferents and efferents. So again, this is going to result in no change in pressure in the glomerulus. So if something's going to result in no change in the pressure in the glomerulus, we're probably not going to be talking about it much more, right? Because it's not going to be a tool really that the kidney is going to use to help us with fluid flux. Adenosine is a vasoconstrictor. This is a bit weird. Adenosine everywhere else in smooth muscle, like in skeletal muscle vasculature, adenosine is a vasodilator. We talked about it as a metabolic vasodilator. Here at the level of the kidney, smooth muscle responds to it to cause vasoconstriction. Easy way to do that, we would just change the membrane receptor type down here. Change the second messenger on the inside to cause constriction rather than dilation. So that's not difficult to do. If I vasoconstricts afferents, not efferents. So efferents don't even have the membrane receptor population there. Okay, so it's only going to affect afferents, which is going to result in a decrease in pressure in the glomerulus. So this is a tool we're probably going to be using to help us with fluid flux. ATP is a vasoconstrictor as well of afferents. So using this tool, we could drop pressure in the glomerulus. And we'll see where some of those come into play in a little bit when it gets important to think about that. There's also endocrine control. Okay, so if we have sympathetic nervous system innervation to the afferents greater than efferents, then you know there's an adrenergic membrane receptor population sitting there, right? So now that you know when the sympathetic nervous system goes to the adrenal glands to release epinephrine, that hormone is going to wander around in the bloodstream and find that membrane receptor population, right? So epinephrine as a hormone is going to do exactly the same thing as the sympathetic nervous system did on a milliseconds to seconds time scale, okay? So no big surprise, epinephrine as a vasoconstrictor, afferents greater than efferents. So we can decrease pressure in the glomerulus with this maneuver. Angiotensin 2, we've talked about it once before as a vasoconstrictor of like skeletal muscle vascular beds. I'm going to put a placeholder for it here, and probably in about 2 days we'll get back to it. It is a vasoconstrictor of efferents over afferents, so a higher membrane receptor population downstream, So we have the ability to constrict downstream of the glomerular capillary bed more than up. Okay, so now we're more in control of volume out of. Okay, we can drop volume out of, which will increase pressure in the glomerulus and help us with fluid flux. Atrial natriuretic peptide. This is a peptide we're going to meet in a couple of days. It's actually made up in the right atria. It has wicked control down here. It can vasodilate afferents and vasoconstrict efferents. So we have two totally separate membrane receptor populations on afferent and efferent, right? So atrial natriuretic peptide comes on board when we massively want to flux fluid, okay? Because if we vasodilate afferents, we're letting more in. Vasoconstrict efferents, we're letting less out. So we get a huge pressure increase in the glomerulus. Okay, so this is really potent in terms of increasing pressure in that glomerulus. So huge control over fluid flux here, okay? And ADH, we've planted ADH in here before. ADH is a vasoconstrictor down here of afferents over efferents. So let less in, let more, or let the equal amount out. We're going to drop pressure in the glomerulus. So even just from that kind of cursory look, we can see what tools the body is probably going to use to change fluid flux when we have a blood volume problem. Because when we have a blood volume problem, we have a blood pressure problem, right? Mean arterial pressure. That's a problem because of, and we can help with a volume by changing volume. We can already probably start to guess what players we're going to bring in to help us with that problem. Okay? So not a surprise, we will be visiting these again in trying to regulate mean arterial pressure. So I'll plant them here for now, let you know of their power, and then we'll bring them in a little bit later when we start to regulate this thing, okay? So that's essentially how we're going to control these afferents and efferent arterioles. The game afoot there is to control pressure in the glomerulus. Okay, so why do we care? So we've been talking about, so here's a little schematic of both of those tube systems that we alluded to on Tuesday. The vasculature system that associates itself with the tubular system of the nephron. Okay, so two sets of tubes running in close association with each other. So here we can see, if we look at the vasculature, we can see our afferent arterioles. So we can see some things that are familiar here. Those afferent arterioles, so we've got flow going in this direction into our glomerular capillary bed. All right, so that's our glomerulus, that's our capillary bed. And then downstream of that, we have our efferent arterioles. Okay, so we've got flow in this direction. And then downstream, so of course we can increase resistance upstream of that glomerular capillary bed. We can change resistance downstream of that capillary bed. And then of course downstream from the efferent arterioles are that second capillary bed, the peritubular capillary. And part of an extension of that peritubular capillary, when it followed along the loop of Henle, in a juxtamedullary nephron, we call that the vasa recta. So again, there's an extension of that peritubular capillary bed that we gave a special name, the vasa recta. Okay, but we will become intimately involved with the vasa recta soon enough. Okay, so that's the vasculature that we've been talking about. It is then in close association with the nephron, right? And so we did talk about the different parts of the nephron. So we're just recapping. Parts of the nephron starts, or this tube starts at the level of the glomerulus. Okay, this Bowman's capsule. So there's this capsule around that glomerular capillary bed, right, and that's where this tube starts. We then, the next part of the tube is referred to as the proximal tubule. They are given different names because they have very different characteristics. So every time we change names, we're changing characteristics fundamentally about what this tube can do. Okay, so we run into the proximal tubule next. And then downstream of that is the loop of Henle. Now there are 3 portions of the loop of Henle. The thin descending limb, and by descending it means it's going down. So let's make sure that we all know that we're talking about a juxtamedullary nephron, because that's the language I'm using here. So just so we're clear, what was the big deal about the juxtamedullary nephron? It had its loop of Henle go way down into that medullary space, and then it had that vasa recta of the vasculature go way down in the medullary space with it. Okay, so the 3 parts of the loop of Henle are the, so if the loop of Henle, if I put it as 3, then subparts of that loop of Henle are the thin descending limb. Oh, I don't know what that word is at all. Nope, can't even make it up. D-S-C, there we are. So if we said 3A, so this is a subpart of that loop of Henle, the thin descending limb, and by descending, we mean descending down through that medullary space. And then it turns a corner, and then it goes back up through that medullary space again. So we've got a thin ascending limb. So hang on, we're talking about this part here is the thin descending limb, thin ascending limb, so let's call that 3B, and that is this part of the tube. Okay, and then as we go up, as we ascend up towards the cortex region, it changes its wall structure, and now it's called the thick ascending limb. So it's still heading up to the cortex, but now its characteristics have changed. So they call it the thick ascending limb. So then up in the level of the cortex now, it's called the distal, so let's call this 3C. It's called the distal tubule. Many distal tubules then converge into the collecting duct, 4, 5. That collecting duct heads down through the medullary space again, okay? This anatomy is critical. And then we talked about how there we would go to the ureter, to the bladder, and then out of the body, okay? So that's a little more up close and personal with our tube. So essentially then, when we're talking about filtration at the level of the kidney, when we're talking about changing, we've just been talking about changing pressure in the glomerulus. We were talking about changing pressure here. Okay, changing pressure in the glomerular capillary bed. So what is the hydrostatic pressure in the capillary bed that we're calling now the glomerulus, right? And we also know that's the blood flowing through there, right? So it's also going to have an oncotic pressure or an osmotic pressure, right? So remember, fluid flux was also dictated by an osmotic pressure created by the proteins that were stuck in that compartment as we filtered fluid out, okay? So if we're going to talk about filtration then, filtration from the glomerulus into where? Okay, the Bowman's capsule, so that's where this filtrate is headed, right? Before we talked about tissue, we went from the fluid, we went from the capillary bed or the lumen of the capillary into the tissue, so we talked about PC and PT. right? Well, here we're headed into a whole new tube, the Bowman's capsule. So there's going to be a pressure now in the Bowman's capsule, hydrostatic pressure in that Bowman's capsule, and there's going to be an oncotic pressure in the Bowman's capsule. So fluid flux is dependent on these four things. So fluid flux across this spot is going to be a delta P, delta pi argument again, okay? So we're going to call this filtration, okay? So they give it a F here. Filtration from the blood to the lumen of the nephron, okay? So lumen of Bowman's capsule. So that word is called filtration. Another word used down here that we're familiar with is reabsorption, right? So once we put it into the tube, if we want it back, we need to bring it back into the bloodstream, right? So there are all kinds of places where they'll show you on this picture fluid moving from the nephron, so here from the proximal tubule back into the capillary bed, right? That would be reabsorption. Note, though, that they've shown it here as a one-step process, like we can just go from one tube to the other. We can't. Here you have to go from you have to go from the nephron out into the interstitial space. You got to go out into that tissue, and then you got to take it from that tissue back into the bloodstream. Right? So this reabsorption is really a two-step process. Their little arrows kind of imply that we can just go straight from this tube to this tube. We can't. There's an interstitial space. There's a tissue space in between. So we have to move. If we want anything back here, we have to move from the lumen of the tubule into the interstitial space, and then from the interstitial space into the blood space. Okay, so that's going to be a two-step process. Other language here that we haven't dealt with before, so secretion. Secretion can occur here as well. So we can get, in other places other than that Bowman's capsule and glomerulus, we can get movement of products from the blood into the tubule. So from the bloodstream into the tubule. So they've got it here as a one-step process. That is not true. If we're going to secrete something, it's got to go into the interstitial space, and then it's got to go into the tubule. And if we secrete something, let's say in this manner, and we don't ever bring it back, so we put it into the tube, and then we don't ever bring it back, it will be excreted. So excreted is a word for right out of the body. Okay, so we're after a little bit of language issues here in terms of what filtration, reabsorption, secretion, and excretion mean. Okay. So essentially, if we back up a little bit, well, let's not do that here. We'll do that in a minute. Okay, so we can now move things into the tubule from the blood space and move things back from the tubule back into the blood space. So here you can already see what's shaping up, right? We're going to do delta P, delta pi, draw that graph, and see when one is greater than the other. You know what's coming, right? So when we move, when we move something into the tubule, that's fine. Boom, we got it in this tubule. Let's say we wanted to reabsorb it back, right? Then we have to actually move that, whatever we wanted, from the lumen of the tubule into the interstitial space, and then from the interstitial space back to the blood, okay? So movement from the tubule into the interstitial space requires that we wander all through these pieces of the tube and look at their individual characteristics. Because every time the name changed, the characteristics changed, okay? So we're going to spend probably the next hour thinking about these different parts of the tubule and how we actually move stuff from here to that interstitial space, okay? So we're going to talk about that. But at the moment, I'd like to talk about how then we actually move once we get it in here. Let's assume we could get stuff in here. What drives movement from that interstitial space back into the blood compartment, okay? So we know that we have a pressure in the tissue, right? So that's just that interstitial space, hydrostatic pressure, and that we have proteins out here, so we're going to have an osmolarity in our tissue space. And the blood that we are flowing by this space will also have, this is the peritubular capillary, right? So we are going to have a pressure in the peritubular capillary because it's a blood compartment. And we're going to, so a hydrostatic pressure, and we're going to have an oncotic pressure in the peritubular capillary. Okay, so if we now assume for the moment that we can move things from the lumen of the tubule into the tissue space, movement from the tissue space back into the blood is going to be a delta P, delta pi argument. We're going to draw the graph and see where they overlap. Okay, so it's the same arguments that we've been doing over and over and over again when it came to filtration and reabsorption. The cool thing about the kidney is it's doing it in 2 steps, right? When we talked about skeletal muscle, there was filtration and reabsorption across 1 capillary bed, wasn't there? So now we seem to have divided it into 2. We've got filtration in one spot and potentially reabsorption in another, okay? So let's see how let's look then at delta P and delta pi when it comes to filtration, and delta P and delta pi in terms of reabsorption to see how the system actually created that, okay? Once we get over filtration and reabsorption, we will spend a lot of time thinking about how did we actually get stuff out into that interstitial space, okay? So we'll deal with that in a minute. But right now, we will look at filtration to reabsorption at the kidney. So again, this is going to be no surprise as to what we're going to be comparing, right? So we're at the level, if we're thinking about filtration, talking about capillary fluid flux, right? Remember at the capillaries of skeletal muscle, we talked about how fluid flux was dictated by a constant, which we never messed with, and delta P and delta pi, and that was our language, pi C, pi T, PC, PT, right? So we've been there before. And we thought, when we thought about fluid flux, it was a delta P versus delta pi argument. So at the level of the kidney, it's exactly the same. It's the language has changed. So capillary fluid flux, we talked about it being represented by J. Fluid flux at the level of glomerulus is called glomerular filtration rate. Okay, so it's the same thing. Fluid flux is dictated by a constant that we're not really going to care much about because we're not going to change it. And a delta P and a delta pi argument. And here our delta P is what is the hydrostatic pressure difference between the glomerulus and the Bowman's capsule. All right, right here. And what is the oncotic difference between the glomerulus and the Bowman's capsule? Blood space, tube space, okay? So if I just give you the numbers, pressure in the glomerulus is high. This is higher. Our hydrostatic pressure in the glomerulus is higher than we've seen it before because we have those 2 funky arteriole sets, right? So normal capillary pressure in the glomerulus is high. Pressure in the Bowman's capsule is about 15 millimeters of mercury. Oncotic pressure in the glomerulus is the same as we've seen it before because it's the same blood that we've seen before. So it has the same osmotic pressures for movement. Remember, osmotic pressures for movement were dictated by the proteins and then the ions attached to the proteins, okay? So, and then in the Bowman's capsule, it's about 8 millimeters of mercury. We don't filter a lot of proteins, okay? So we filter a lot of stuff here, but we don't filter a lot of proteins. Okay, so if we then just play the equation game here, So if we look at our delta P, 60 minus 15, our delta pi, 28 minus 8, we get 45 minus 20. So 45 minus 20, we get delta P greater than delta pi. Okay? This was a scenario we set up before, which would argue we get filtration, right? Now what the kidney did is set pressure higher, right? So we talked about this this hydrostatic pressure now is set high. Let's label our graph here. And then we have our glomerular capillary bed. And we know that we have to have capillary pressure drop across the glomerular capillary bed or there'll be no flow. So the same rules apply here, right? So we have to have PC or PG in this case dropping as we go, right? So that's our delta P argument from before. And then we started out with, so that started out at 60 and it drops across the glomerular capillary bed. Our osmotic pressure in the glomerulus starts out at 28, right? So 28. And because we have a lot of fluid filtering, so we have larger slit pores here, we have a lot more fluid filtering across this capillary bed. We actually have, we actually concentrate the blood a little bit as we go, right? So we are pulling out fluid, leaving those proteins behind, so the blood becomes a little concentrated as we go. So this is something that was a little bit different from our skeletal muscle capillary bed. Okay, so this is really our delta pi as we go across that capillary bed. Cool, though, is because this tissue pegged the delta P higher than normal, delta P and delta pi never cross. So there's no spot across the glomerular capillary bed where delta P is less than delta pi. So we filter across the entire capillary bed. Okay? So nothing magic here other than setting a capillary bed between two sets of arterioles and jacking the pressure up a little bit. Okay, so no special mechanism to create a filter, right? It's just that now, I guess in terms of our language, from our skeletal muscle examples, we would be filtering across this entire capillary bed, which is great. Because what's the kidney? The kidney is a filter. Okay, boom. Higher pressure, we've just made a filter. Okay, so we don't have to make something super fancy. We just use the same rules, the same basic rules in terms of how the system is set up, and change one of the variables. So at the end of the day, let's write it in words. We filter across the whole capillary bed. And of course, filtration across the whole capillary bed, net movement from the blood compartment into the Bowman's capsule across the entire capillary bed, okay? So this portion of the relationship between the blood vessel and the lumen of the tube is that we have filtration across the entire capillary bed. So no filtration. So that's why we just have an arrow going in this direction and not one coming back. Okay, there's no reabsorption happening at this level of the capillary bed, or at that capillary bed at all. All right, so then let's think a little bit. We got 2 minutes. Okay, where are we here? So now we've got all kinds of filtrate in here, which is great, because we're going to need to work with that once we think about this tube. And then once we think about this tube, we will actually see how this tube then moves all kinds of stuff from the lumen of the tube into that interstitial space. Okay, but that's going to take us about an hour to get through that. But while we're here, let's then think about how we get things from that interstitial space back into the blood, reabsorption. Let's think about that second part of that reabsorption argument while we're here. Because it's really just going to be the same, it's going to be the same set of arguments. Okay, it's going to be a delta P, delta pi argument. But you can see what we're kind of setting up here. If we have filtration across one whole capillary bed, one would hope we would have reabsorption across the other capillary bed. Okay? Let's see how it does that. So if we think about reabsorption, so this is into the so we're going to be thinking about reabsorption into the peritubular capillary bed, so that second capillary bed downstream of that glomerulus. If we think about just a little bit about where we are, so here's my my lumen of my tubule. Okay, so this is our nephron. And we've said, okay, we're just going to assume that stuff comes out. And again, we're going to spend an hour thinking about how stuff comes out. Right? And then we have this interstitial space. And then we have that peritubular capillary. So this is just that two-step reabsorption that I talked about. This is our peritubular capillary. So this is our blood space again, right? So let's leave this discussion for a little bit later and know that in our interstitial space we're going to have a hydrostatic pressure. And we're going to have an osmotic pressure. It's not really different than those that we've seen before. Peritubular capillary has a hydrostatic pressure in the peritubular capillary. (13 millimeters of mercury). Okay? Remember, pressure is dropping across the capillary bed and across any piece of tissue. When we go from the artery side to the vein side, we're heading towards 7 millimeters of mercury, right? We're heading towards mean circulatory filling pressure. So we're almost there, right? We got a low pressure capillary bed here. But if we think about delta P, this will result in movement from the peritubular capillary bed into the interstitial space, right? That does not look much like reabsorption, right? But the interesting thing is, as a result of taking so much volume out at the Bowman's capsule, we concentrated the blood, right? This blood is about to head into the peritubular capillary. So you have concentrated blood that is now flowing through the peritubular capillary. So this number is going to be a little bit different than ones we've seen here. So our delta, or our osmotic pressure in the peritubular capillaries is about 32 millimeters of mercury. What does this mean? This means the blood is very concentrated in this space. How does that affect water or fluid movement? Water moves to dilute. So water is going to be drawn into the blood space because it's so concentrated, right? So in terms of hydrostatic pressure movement, we have a much larger drive to move fluid osmotically than we do hydrostatically, okay? This is a classic, back to our classic fluid flux equation. So if you dig equations, we're thinking about capillary fluid, our capillary filtration constant, which we're going to leave constant, and we're going to think about delta P, and delta P was hydrostatic pressure in the peritubular capillary minus hydrostatic pressure in the tissue, delta P, and delta pi, oncotic pressure in the peritubular capillary minus oncotic pressure in the tissue. Okay, delta pi. Sub in the numbers that we care about. 13 minus 6, minus 32 minus 15. Okay, so we have a delta p of 7, a delta pi of 17. Okay, so this is now showing us, through an equation, if you dig those, that our delta pi is greater than delta p. Okay, and that was the scenario in which we saw reabsorption. So if we graph that like we have before, now we're talking about the paratubular capillary bed. Remember where we're at? So now we're going to have the afferent or the efferent arteriolar side, paratubular capillaries to our venous side. If we have in the blood a hydrostatic pressure of 13, so this is pressure millimeters of mercury. Okay, if we started out at 13, we know pressure has to drop across this capillary bed towards venous pressure, right? So across this capillary bed, we should actually see delta P drop. We need flow. Flow has to happen, right? So we're going from 13 towards venous pressure 7, okay? Oncotic pressure started out at 32, and as we move fluid, as the oncotic pressure draws fluid from the interstitial space into the blood compartment, it will start to dilute, right? So we should fully expect our oncotic pressure to drop as we go. Back down to our normal blood osmolarity. But at no time does delta P ever get greater than delta pi. Delta pi is always greater than delta P. So in the language that we've been talking about before, we would have reabsorption across this entire vascular bed. Okay, so what the kidney did is, based on changing hydrostatic pressure and changing oncotic pressure, is it separated out filtration and reabsorption into 2 capillary beds, and it stuck a nephron in between. Okay? That's essentially what we did here. Every other piece of tissue we looked at had filtration and reabsorption across the same capillary bed. Right? Here we tweak hydrostatic pressure up in one capillary bed, tweak oncotic pressure up in another, and split filtration and reabsorption into 2 capillary beds. Stuck this tube in between. Now we have to figure out what the heck this tube is doing. Okay? So, I'll write this out. So we're always reabsorbing. All right, so the kidney has a filter. Okay, cool. Let's take a 10-minute break there, and we'll get back to thinking about that tube, because that's what we'll spend the next hour or so on. Okay, let's get back at it. Now that we've had time to mull this stuff over a little bit, I've thrown a lot at you here today and asked you to remember stuff from like 3 weeks ago in terms of filtration and reabsorption. What questions can I clear up? Are there something that I've gone over here that you're just like, what are you doing, Coral? Why did that line go up? Why did that line go down? Anything bugging people about filtration, reabsorption, argument split into 2? Hydrostatic pressures, oncotic pressures. We all comfy? Yeah? Yes. Yeah. Sure, yeah. The difference between filtration and secretion, exactly where it's happening. Yeah. And the process by which we will do it. So if I'm going to do all my fancy filtration here at the level of the Bowman's capsule, if I'm going to move something from downstream, and we will see this, it's because it's going to be a nature of the tubule issue, I'm going to have to actually have a transporter for something to move it from here to here, and then a transporter to move it from here to here. So secretion is actually more of a membrane receptor dependent process than it is just a delta P, delta pi argument. But we'll get to that when we actually look at the nature of that tube. But it is another way to move something into the tube. So that's an important process for sure. Okay, any other thoughts there before we move on? Because now what we need to do is spend some time. We told you how we are going to get stuff filtered into this tubule. And then we talked about, okay, if we could get it out into the interstitial space, we know we can get it back into the bloodstream. So now the game afoot is, how do we get it from the tubule into that interstitial space? Okay, so now we're going to be dealing with this arrow here, essentially. Okay. All right, let's head into that tubule for a little bit. So just before we head there, little thought about the kidney, little thinking about if you were to design this system or if you were to make it, how would you do it? So we have to build a filter, right? So there's two ways to build a filter, right? I've got this blood, and we talked about how the kidney, one of its jobs is to filter nasty bad stuff out of that blood compartment that we don't want and put it in this other tube, connect that tube up with another tube that will get it out of the body, okay? So that's why we're switching tubes, right? So that's why there's a whole tube system put in place here. But when we talk about filtering, there are two philosophies of doing that, right? I can set up at the Bowman's capsule, so that Bowman's capsule is just a layer of endothelial cells. We could set up that Bowman's capsule with transporters for every bad thing in the body, right? And then as that blood flowed by, we could just be plucking that bad stuff out, right? So the problem with that design, it's a great design, the problem with that design is people eat weird stuff, okay? And that would mean that the body would have to predict every weird piece of stuff you are ever going to ingest in your entire life. And if it can't predict that, it can't get rid of it. People eat bicycles. There's weird stuff. People eat all kinds of, if you look at the Guinness Book of World Records, what are they doing, right? So you'd have to have a bicycle receptor in there to move that bicycle from the blood compartment into the lumen of the tubule, okay? People eat weird stuff. And it changes over time, right? So the body would have to be pretty rapidly adapting to keep changing that layer to move weird stuff from the blood compartment into the Bowman's capsule or into the second tube to get rid of it. So that design has a flaw. So the second design, which is the way that this kidney works, is it filters everything and then sets the wall of the tube that we just put it into, designs that tube to take back stuff we want. And now by default, anything that we filtered that we didn't want goes out, right? So the body is really good at knowing what it wants, right? So what we're going to do then is we're going to go up and down this tube, and we're going to be seeing that along the length of that tube is built to constantly take stuff out we want, and then by default, everything else goes, okay? So the philosophy of this filter is filter everything and take back what we want, okay? So we're going to wander along this tube, seeing where the body put different mechanisms to take back what we want, okay? And by default, if we didn't take it back, it's stuck in this tube and then essentially out, okay? So your bicycle can be gone without ever having a bicycle receptor, okay? So we need then to wander along all these different parts of the tubule that we named, all have different characteristics. Those spots of the tube are taking back different things that the body wants, okay? So the game afoot then will be to look at the different bits of the tube, the tubule, and see what the body wants. Okay, so we're going to start at the front, the proximal tubule. Okay, so what I've got here is the lumen of the tubule is over on this side. All right, so this is the stuff that we just filtered. Right, so we're at that glomerulus, filtered across that Bowman's capsule, because delta P is always greater than delta pi. Boom, we've got fluid in the lumen of this tube. And then there's the tube is made up of a single cell layer with tight junctions in between. This is our interstitial space, so this is the other side of that cell layer. Okay, and then I've got a representation of the vasculature, another set of endothelial cells, the vasculature on the other side. Okay, so essentially we're looking at what is this, the nature of this wall. Let's do it, yeah, this wall here, and how it's going to relate to the vasculature that's going by it, okay? So this is indeed the vasculature. That's our peritubular capillary. We've already then discussed how we move stuff from that interstitial space into the peritubular capillary. That was all reabsorption, right? Where delta pi was greater than delta p. So we got that bit nailed. And now we're thinking about how are we going to move from the lumen of the tubule into the interstitial space. So I've got 4 cells drawn here. These are not meant to depict 4 individual cells. These are just meant to depict different processes. Okay, each cell could have each of these processes on them. When specific cells are present that have specific mechanisms, I'll let you know, because there are these cell types down here, like intercalated cells have very specific mechanisms on them, et cetera, et cetera. But I'll flag those when we go. So essentially, we have asymmetrical cells. So the whole kidney is built on this common theme throughout physiology of cell asymmetry. If I want to move something from compartment A to compartment B, I just build a cell that has decorated on one side of it differently than what's decorated on the other. So I can put glucose transporters on the luminal side of the tubule of the cell. Glucose transporters that, a symporter, right? So we're moving it along with sodium, so this goes way back into human phys 1. I can move glucose in, and then I put a facilitated transporter on the other side, and I can move it out into the interstitial space. Boom, there it is. Once we know we can get it there, we know we can reabsorb it. We've seen how we do that, okay? So now we just set up all kinds of symporters for stuff we want. Glucose, high energy store, we need that back. Okay, so we will have massive numbers of glucose transporters down here to get every lick of glucose that we filtered. That's what we wanted to get it back. Okay, so no glucose usually makes it out of the proximal tubule, unless you're diabetic. If your blood glucose is way too high, you get a huge amount of glucose in the lumen of the tubule. These are just transporters. They have a transport maximum. They could all be working. So we can then actually lose glucose through having too much on this side, but under normal healthy conditions, we will get back all of the glucose we filtered. So this is the body getting back what it wants. So now there's just going to be a massive number of symporters in the proximal tubule. Symporters for amino acids that got filtered, symporters for things like phosphate, lactate, bicarb, all kinds of stuff, okay? This is the body getting back what it wanted. Put it in the interstitial space, and then we know we can reabsorb it back from there. Important here, too, is that water is moving then. If I move all kinds of stuff like glucose into the interstitial space, water will be drawn to dilute, okay? So really critical is that water moves by osmosis, right? So we can get water movement secondary to the movement of all this good stuff. So water moves by osmosis. There are also water channels down here. Anybody know what these water, have you heard of these water channels before? Do you know what they're called? There's like water channels in these membranes. Have you heard of them in biochemistry? Aquaporins. Which are weird. Fundamentally do not understand an aquaporin. Okay, think about why. Like, so how did we, even in Human Phys 1, and you talked about water movement across a membrane. Did you talk about how easy it was for water to move across a membrane? It's weird because it's non-lipid soluble, but it can move across that lipid membrane, and the thought is because it's a small kinetic molecule, water moves across membranes just like the membrane isn't there, right? That's how we've been treating water movement. And then we say, all right, now I'm going to help water movement by putting in an aquaporin. Well, why did water movement need help in the first place? If it moves through a membrane, like the membrane isn't even there, then why you put all this energy into building a protein, sticking it in the membrane to make it more permeable? So I have no answer to that. I'm just going to leave that one for you to get to grad school, figure it out. I'm going to give you about 10 years, circle back and let me know, right? Because I do not fundamentally understand. There's something fundamental we don't understand about water movement, all right? We should not need an aquaporin. Proteins are expensive. We've got to build them and put them in a membrane, and then we've got to keep cycling them, taking them out, putting new ones in, taking them out, putting new ones in. Why would we do that? That's expensive if we didn't need to. But again, we talked in Human Phys 1 about how water moves across a membrane like it just doesn't matter, okay? So we think we're big and bad because we can like clone a sheep, but at the end of the day, we have no idea how water moves through a membrane, okay? Really basic stuff, really critical to knowing how we work. So I'm going to place it there, but I'm not happy about it at all, okay? So 10 years, get back to me. So if water is moving through osmosis and through these water channels, and I guess the interesting thing about water channels, again, you can tell they bug me. If I'm going to have a water channel on one side, I have to have a water channel on the other, right? So now I've got to decorate both sides with water channels. Damn, that's work, okay? And we'll actually use that as a tool. There's a hormone that increases the number of water channels. Drives me crazy, but we'll get there in a minute. Okay, so water can move by osmosis, so we can move through those tight junctions, or we can move through with aquaporins. That's fantastic. And so then things that are dissolved in that fluid, right? So there are things soluble in there. There's our ions, sodium and chloride and potassium, CO2, calcium. We've got all kinds of stuff that is solubilized within that water. Okay, so if we think, let's do this argument here. I talked about those first, and then I talked about water movement, and then we get diffusion of ions. So these are things we want. Again, this is the tube taking back what we want. We can get diffusion of high concentration to low. Obviously, sodium will be moving down its concentration gradient, right? We can get diffusion of some of these ions through, but we can also get them through something called solvent drag. So when water moves, it's going to pull solutes with it. Okay, so at the end of the day, we've gotten back glucose, amino acids, lactate, phosphate, all those types of things. We have symporters for those. And then as water moves down its concentration gradient by osmosis, we're also going to be dragging along other ions and things that are dissolved in that water. Okay, so you can already see things that we've been talking about over and over and over again that the body uses at a fundamental level. We filtered it, and now we have a way of getting it back. Okay? And then we have some other special mechanisms for special ions. So for instance, we have some transporters set up to deal with bicarb and hydrogen ion. So we'll have an asymmetry cells that have antiporters for bicarb or hydrogen ion, depending on what we want to do. I'm going to plant these here. I'm going to plant these here, and we're going to come back to these when we talk about acid-base balance. Okay, if you have pH is a regulated variable. Hydrogen ion is a regulated variable. So in here, we're still trying to regulate mean arterial pressure, but laid in here is also other systems to help us regulate hydrogen ion. And we're starting to see them here. So I'm just going to plant them, and then we're going to come back to them when we think about regulating hydrogen ion. Another part of regulating hydrogen ion is something called ionic capture. I can get rid of hydrogen ions in the form of ammonia, move them across an asymmetrical cell onto the luminal side. That ammonia can trap or capture a hydrogen ion. The membrane isn't permeable to ammonium. All right, so this little maneuver trapped a hydrogen ion on the luminal side. We didn't have any way to bring, we don't want ammonium, right? Or else we'd get it back. We don't want ammonium, but we trapped a hydrogen ion in there with it, and we can excrete that out now. So this is a little trick for regulating hydrogen ion. I'm going to leave it there. We're going to circle back to that, okay? When we start regulating something else. And then there's these different transporters in the membrane to deal with secretion. So this is that secretion argument where, let's say we did get through the peritubular capillary, we still have stuff in there that we don't want. We've got to have ways of secreting that out. So when we think about moving something from the peritubular capillary into the interstitial space, and then we have to set up an asymmetrical cell to move it from the interstitial space over to the lumen of the tubule. Okay, so we just set up things like there are anions that we want to get rid of. So we have antiporters for anions to get that anion into the cell and then get that anion out. Right, so we can move that anion into the cell, move it into the lumen of the tubule, and if we have no way to set up, no transporter set up to get that back, boom, it's gone. Okay? So things like bile salts. So these are things that would normally be occurring in the body. Second messengers, we can also get rid of those. Things that might not normally be in there, so weird stuff people take in like penicillin. Okay, so these are broad categories of transporters to get rid of stuff that we would normally experience, but then also helps us get rid of stuff that we wouldn't normally experience. Same is set up for cations. Things we would normally experience, like this is how we get rid of. Remember, we've been increasing epinephrine and norepinephrine in the body for a long time, right, to help us with how to regulate mean arterial pressure. But we got to get rid of it. So the kidney helps us with that. So epinephrine and norepinephrine can be moved into the interstitial space. And then we've got cation channels, transporters that can move it over into the tubule. Good as gone. Things that we might expect like epi and norepi. Things that we might not expect like morphine can be moved out this way. Okay? And as long as we don't have another way to move the morphine back, we're not going to keep it, right? So that's an indication the body didn't want that. Okay, so at the end of the day, that's the proximal tubule in a nutshell. Okay, we'll talk more about this on Thursday, next Tuesday, and tomorrow is a problem-solving session. Okay, have a good day.